Today, we'll be discussing contractile vacuole complexes in algae. Contractile vacuoles are organelles which occur in many protists and some freshwater sponges. They are defined by their behavior of slowly filling with fluid and periodically expelling this fluid from the cell. Contractile vacuoles form one part of a more extensive organelle, the contractile vacuole complex (CVC). CVC typically includes the contractile vacuole, a system of membranous elements called the spongiome, and a permanent pore supported by cytoskeletal structures, which ensure the expulsion of the fluid at the fixed site on the surface of the cell. Contractile vacuole complexes have usually been implicated in the osmoregulatory competence of the cells. Distribution of contractile vacuole complexes in algae. CVC are found in most type of the algae which have species living in freshwater environments such as volvocales, xenophytes, euglenoids, cryptophytes, chrysophytes, then haptophytes, raphidiophytes, and some heterochloride flagellates. Contractile vacuole complexes have not been identified in the stigmatophytes, marine silicoflagellates, and dinoflagellates. Algal flagellate cells typically contain one or two contractile vacuole complexes. Most frequently, they lie in the vicinity of the flagellar basis and discharge their contents at fixed points in the surface of the cell. Discharge of vacuolar contents form one part of a recurring cycle of behavior. This incorporates two major phases: filling or diastole and expulsion or systole. At the light microscopical level, filling appear to occur by the fusion of small accessory or contributory vesicles. The term contractile vacuole is applied to the largest of the vesicles. Diastole usually terminates with the rounding up of the vacuole. Simultaneously, the contractile vacuole becomes slightly smaller in diameter, and these events have been interpreted as indicating the contraction of elements intimately associated with the vacuole. During the systole, the vacuole appears to fuse with the plasma membrane and disappears from the view as the content are lost. The ability of the algal cell to adjust to the changes in the salinity of the medium is an important aspect of the physiology of cells. In cells with wall, this osmoregulation is accomplished with the aid of turgor pressure, whereas in naked cells, it is accomplished by means of contractile vacuoles or regulation of the solute present in the cell. In the later cases, cells increase the internal concentration of osmotically active molecules and ions when the contraction of dissolved solute increases in the external medium. Likewise, the internal concentration of such molecules decreases when the concentration of dissolved salt in the external medium decreases. Most algal flagellates have two contractile vacuoles in the anterior end of the cell. A contractile vacuole will fill with an aqueous solution, means diastole, and then expel the solution out of the cell and contract, means systole. The contractile vacuole rhythmically repeats this procedure. If there are two contractile vacuoles, they usually fill and empty alternatively. Contractile vacuoles occur more frequently in freshwater than marine algae, and phenomena that gives credence to the theory that the contractile vacuoles maintain a water balance in the cells. The algal cells in the freshwater have a higher concentration of dissolved substances in the protoplasm than in the surrounding medium, so that there is a net increase of water in the cell. The contractile vacuoles act to expel this excess water. An alternate theory on functions of the contractile vacuoles is that they remove waste products from the cells. The contractile vacuole of the cryptophyta are characteristic of the algae. In the cryptophyta, contractile vacuoles occur in a fixed anterior position to the flagellar depression. Just look at this diagram. This first part, this is flagellar depression, and contractile vacuole is going to form here. So, in the beginning of the filling phase, means diastole, there is no distinct contractile vacuole. You can see here, this part. Only a region filled with the small contributory vacuoles you can see here. Then these vacuoles fuse to form a large irregular vacuole, which subsequently rounds up. The contributory vacuoles destined to form the next contractile vacuoles now appear around the plasma membrane of the flagellar pocket, and you can see here this is contractile vacuole. Now this contractile vacuole now appear around the rounded contractile vacuole, and vacuole then fuse with the plasma membrane of the flagellar pocket. Here you can see this is flagellar pocket, and contractile vacuole fuses with this. Then discharge. Then next discharge. This contractile vacuole discharges its content outside the cell. Right? So the area of plasma membrane that fuses with the contractile vacuole does not have periplasm, and this area is instead bounded by microtubules. The membrane of the contractile vacuole is recovered by the cell as small vesicle with the electron dense coat, and then membrane components are reutilized by the cell. These vesicles plus the contractile vacuole occur in the spongiome, or means area around the contractile vacuole, and freshwater algae. Contractile vacuole cycle lasts for 4 to 16 seconds, whereas in the marine species, this cycle can last up to 40 seconds. The algal flagellate use a combination of contractile vacuoles and osmoregulation to control the water content of their cells. In some chrysophytes, here the internal level of isopyridoside OD galactopyrinoside 1,1-glycerol is proportional to the external osmotic value as long as the external solute concentration exceeds 75 mosm. Below this external solute concentration, the influx of water into the cytoplasm is counterbalanced by means of the contractile vacuoles. Thank you.